video first. Share the screen. Yeah, that's good. And Dr. Chu, if you are ready, and then we can get started.
Yes, I'm ready.
Okay.
Yeah, last time I was using this set of PowerPoint, which is the implantation in soft matter. Actually, the title is misleading, and however, I want to give an overview about the implantation facility and the rough application. And before I get into the semiconductor, the semiconductor implantation probably will have to wait until the next session, next week or some other time. So first, you're all familiar right now. Most of the students you're working in this lab is we have the high energy implantation, which is in the upper left corner. So the long beam line will give you the MeV or several hundred keV type of implant. On the right part, upper right, this is shown the ion source part, which is I'm sorry. So there's a 200 keV ion implantation machine. That machine is idling, and actually 200 keV is a very broad end region. And since we were using the MeV most of the time, so this machine is not working. And also on the bottom picture, there's a cluster beam machine. The cluster beam machine is very powerful and very useful, despite of its being small, and so the function is quite different. First, let's look at the energy. The energy is 30 keV. That's low energy. The cluster means not a single ion, not a single atom, but rather it's a cluster of atoms. So now let's say the cluster of atoms could be a few hundred, could be a few thousand. Actually, the ion source is including a high pressure gas bottle. Let's say argon. So argon is the one we frequently use. If you have an argon gas, which compressed in a steel bottle, if you mechanically leak the argon gas into the vacuum, then what happens is then this gas will see the vacuum and immediately condense into droplets, almost like a fork machine. You like a hairspray. You spread the argon molecule into the vacuum, then the argon immediately frozen into a small droplets. So the droplets having the size of a few hundred to a few thousand atoms. Then you spread some electron on top, so those droplets become a charged particle. Now you put in 30 keV, then they start really accelerating. Then you set up some kind of quadrupole. You can select the mass. Let's say we select 3,000. That means 3,000 atoms, 3,000 argon atoms. Droplets will be preferentially go through a acceleration stage. All the other either get distorted or get steered away from this beam path. So now you select the droplets, select the cluster. 3,000 atoms with 30 keV. That means each atom will only have 10 electron volts. So this is almost like you have a spherical shape, droplets with 3,000 atoms moving together, and they only have one electron missing. That means it's singly charged. And so with 10 electron volts equivalent per atom. So this is very, very slow, very gentle. It just go down the vacuum until it reach to the target. Next one. Next graph. Okay, here we were showing when the cluster beam hitting the sample, what happened? The most important function is to smooth the surface. I remember last times at the end of the talk, we're talking about the atomic force microscope can measure the smoothness. So the smoothing the sample sometimes is very useful. But this machine will smooth the sample down to the nanometer, down to 1 nanometer. So on the upper left figure, this is a film of superconductor. It's a single crystal film. So when you deposit a single crystal on a substrate, the film, although it's a single crystal, but due to the nature of the crystal growth, so the surface is very bumpy, like an orange peel, it's not smooth. So if you are normally incident with 30 keV argon, what happened after a while, it was smooth into the upper right figure. You can see some of the chunks, like a copper oxide mountains, get eroded and getting shorter. And then if you continue to erode, go to the lower right, so the surface is all the features removed. So it removed down to 1 nanometer. So that means the surface is only 10 angstrom up and down. So this becomes extremely useful for making device, for doing some other type of study. So the lower right is a magnification about one of the figures. And so showing the very nice surface feature. This beam amorphized the layer, only the surface portion. It amorphized roughly about 5 nanometers. However, by increased the temperature, you can regrow into single crystal and make this film useful. And this is one method trying to make it squid. That means a superconducting quantum device and so on. So the reason I mentioned here is that implantation could involving atoms, could involving MEV, but it could also involving KEV. It could involving dimers, trimers, and also cluster atoms. All right, let's go. Let's skip many of them, and then let's go into the. Let me see in which figure. Okay. Into figure 23. All right. This is one of the university at the Turkey. This university, their research specializes in ion implantation in large equipment. So you can see their target chamber is actually can housing several people. So the student can actually get into the chamber trying to making modification and so on, modifications. So the implantation here, in this case, is really for industrial application. For example, you can put in a airplane turbine machine into it, and therefore the implantation will be on the turbine, those kind of very tiny, very sophisticated shaped turbines pieces. So in this case, the implantation of nitrogen, if you implant nitrogen on the metal, it makes the metal very hard and very, very hard. And so it's a strong. So this is one application I'd like to mention, since I want to say how useful is implantation. So this is one way. Also, you can use for making all the cutting tools in a machine shop. You have driller, you have cutters, you have lots of large, many small pieces, or even the steak knife can be implanted and make it very, very hard. So the implantation is one thing. The next one. So this is showing, I apologize for all the language here. It's in Turkish. So I don't know how to read and so on, but it doesn't matter. So the important part is the target pieces could be extremely big. Okay, and therefore what they do is they using a nitrogen plasma. Therefore, the plasma is occupying the whole chamber, and the plasma is the ionized nitrogen. This ionized nitrogen will be constantly bombarding your sample. Therefore, the sample will be soaked, will be implanted with nitrogen, but the nitrogen will make the material property change. Next view graph. Here I'm showing another example. Although we are not working for this in this area, by showing the example, we broaden your scope and let you know the implantation a little bit better. First, let's look at the picture. The picture here is showing a artificial bone. This is a artificial. It's equivalent to our lake. Okay, our lake connect to our connect to our body. You can see the head. The head is almost like a spherical shape, and then you can see the stem. The stem is like a leg bone. Okay.
Dr. Chen, I think you haven't gone to the next slide. He is describing the next slide.
Yes. Thank you.
One, right?
Yes. Go next view graph. Okay. So this one is showing the implantation in polymer. Okay, in polymer. So this actually is one of the dissertation work of my students. Because the implantation having electronic energy loss, therefore there's a lot of ionization. So if you start with the polymer, then the polymer will be ionized. There could be bond breaking, and also cross-linking, and the radiation damage and repair, and so on. So this is a very interesting part. So this student got his PhD under me many years ago. I bring his thesis topic here. It's just try to broaden your scope, let you know what's happening. So in this case, because the molecule is very complicated, organic material, so the density will change. It will change the hydrophilicity, hydrophobicity, and also it could forming new compounds and so on. The next view graph. So this polymer has a very long chain, the CH, CH2, and so on. So once you bombard it with proton, and proton is hydrogen ion, so once you bombard with that, you can break the bond, or you can cross-linking, you could making different molecules. Next one. Next view graph. Next view graph. Okay, and next view graph. Somehow this is, we were not in the same set. Okay, so I'll keep going, keep going. Keep going, go to the 24. All right.
24?
So, yeah, right here.
Oh, 34.
34, I'm sorry. So So this one, what we were doing is the application is quite interesting, because once the patient goes to the hospital, quite frequently they are giving them the IV treatment. IV is you have a bag of liquid, so the liquid contains the drug or contains the nutrition, then trying to really feed into the patient's bloodstream. So the bag itself went through the neutron radiation because they want to really kill the germ. But sometimes the neutron is a overdose. Therefore, they could be having very tiny holes on those kind of bag. But this is dangerous because if the bag is leaking, and this could be very bad. So what my student will study is trying to study the neutron damage and using proton to damage it and trying to study the cross-linking and the molecular change, the reliability and the integrity of the bag. So that was a part of his study. So let me just sidetrack a little bit. So what we found out is if the high molecular weight material, if the bond is broken, then can we anneal it? So the answer is yes. If you anneal in hydrogen end-beam, you actually can repair the bond and try to make the material stronger and so on. So this is one of the byproduct we got out of his research. Let's continue. Do you, I'm sorry. Could you go to Yoga 25?
Yeah, this is the 25, right? Okay.
25. Go back to 25.
Okay.
Thank you. What I want to show here is the medical application. And the photograph here is showing a hip joint. The hip joint is really a leg bone. This is artificial, made of high molecular weight polyethylene material. So this leg bone, artificial leg bone, contains a head, and also just like a ball joint, and also down there they have the leg, okay, the stem. When the old people broke their hip joints, they need to really making a new set. Okay, so this is a set, but the friction wear on the head and the joint there is really pretty bad, because once the patient putting a new joint, they feel happy because locomotive, all the motion is returned to normal. However, after using 6 years, and the head becomes due to this rotational motion and the rub, and they become consumed. Therefore, they can actually become wear off. 6 years later, the patient start complaining. So the typical process is they call the patient back, cut him open and give him a new set. Very painful and very expensive. So my friend doing the eye implantation, Mr. Jim Williams at the Oak Ridge National Lab, he said, well, how about the modification of material? Suppose he using high energy proton irradiation on this head, and therefore the head become much harder, much stronger, and much smoother. Therefore, it's not going to be wear off easily. So this turned out to be a very important patent, made lots of money for the national lab, and also enriched himself. So used to be 6 years, patients start to complain. Right now, you're less 60 years, order of magnitude improvement. As you know, the old people cannot live another 60 years, so that means a lifetime guarantee. So this is really a very, very good implantation study on that. Okay, let's go to the, let me see where I want to go. Okay, I want to get into the hydrophilicity type of study. So this is the 37, please. I go to view.
Okay.
Right here. Okay. So here, this is also a dissertation by Iran's work. The contact angle is changed because the surface chemistry is changed due to ion implantation, and therefore you can really make the surface. The surface here is a solid. The water droplets on the top. But measure the angle of the water droplet, you can tell the solid surface is hydrophilic or hydrophobic. By the time you reach to the third figure from the left, so this is a highly hydrophobic. That means the water doesn't stick to the, doesn't stick to it. So then that's very good using your bubble method. This trying to make the surface having different hydro property will be very important to a biophysics study. Let's go to the next. Okay. The other type of application here is just for your awareness, and also if students are interested, they can also get research in this area. Is using our implantation to form antibacterial soft material. Okay, they can they can form in guide tube for nerve regeneration. They can pattern the cell, or they can forming a platform for the stem cell research. They can form artificial neuron and so on. So, because whenever we talk about implantation, people say, well, semiconductor becomes so recognized and also so applied in the industry. So we know that the implantation method, I mentioned about the turbine, I mentioned about some of the Turkish work and the drill and the machine tools and so on. So the bio part is still a very, very open area. Not too many people study it and so on. So I do want to mention the next one, please. Okay, on this view graph, they are using a silver implantation into a sample. This sample is a glass plates. Okay, so those kind of very, very small, I would say it's really like a small pancake shaped container. It's in biochemistry or biology, they do this a lot. They culture the bacteria, they culture the cell, then trying to study reaction and so on. So in this case, the silver implantation or sample, somehow the E. coli, you put the E. coli in there, you put the E. coli there, then E. coli start to multiply.
就可以了。它就是因为两个螺根有的时候是那个。
I beg your pardon?
I'm sorry. In the next slide.
Right. No, not yet. Okay.
I have a phone call. I'm sorry.
The different.
您说一个就够了。
Yeah, one can calculate it. One can count the E. coli population or their colony and by the density of those particles.
意思是说绕时间干。
Right. Then on the fourth quadrant, the figure on the left bottom is showing after 24 hours, all the E. coli die.
那好啊，那要是那个被放到后面，就自由度就很大了。
Right. Somehow that indicating the silver having the property about antibacterial properties, and so on. So these are quite interesting. Next view graph. Okay, here here's another example. Okay, so if you don't have this antibacterial silver implant, the bacterial start grow just as is. Okay, but if you have implantation of silver, then this is right now this is the cotton fiber. Okay, you're using a cotton cloth, and you implant the cloth. Then this cloth turn out to be antibacterial. So I remember some years ago, a small company even thinking about making Q-tip. The Q-tip is a cotton fiber implant with silver. So this way, your Q-tip having the property about antibacterial, which is good. Okay, for certain purpose. Let me keep going.
Professor, I have a question.
Yes.
About the previous two slides. What is so special with silver that it reduces bacteria?
Right. E. coli is a bacteria.
Yes.
Okay. And also in the mud, we have lots of bacteria. So the bacterial is different from germ. It's different from germ, also different from virus.
Yes.
But bacteria is a cell which multiply themselves.
Yes.
What happened is, let's pick the E. coli. The E. coli, but if you pick one E. coli under high magnification, it looks like a sausage.
Yes.
It's had the shape of a sausage. Then through taking the nutrition from surrounding, this sausage will grow longer and longer. And also pretty soon, the center portion will get narrower. And then suddenly one sausage become two sausages. Okay, so this is the way they multiply. One become two, two become four, four become eight, and so on and so on. Then pretty soon they become a colony. Okay, so then that's the E. coli. That's E. coli. Okay, so of course the growth requires nutrient. The nutrient has to come from outside, and therefore when a patient, but we get lots of E. coli in our intestine, and therefore the intestine it can handle that. And also we also expel the E. coli when we go to bathroom. But somehow if the E. coli get into the blood, which I had this problem, that's why I'm interested in this topic, that the E. coli get contaminated to my blood. Then inside the blood, the E. coli just feel so comfortable because there are so many nutrients. So they start taking the nutrient and then start multiply inside the blood. But once the E. coli reach into certain population, then the blood will be deprived with neutron and deprived with oxygen, with other things. Then when the blood flow to the organ having insufficient nutrition and insufficient oxygen, then the organs start dying. So this is the danger about E. coli in the blood. Okay, eventually this disease is called the sepsis. Septic tank is one of the toilet. When we go to the toilet, we flush. That part of it is called septic tank. So septic tank is very, very dirty. So the disease, because sepsis is, that means your blood system, almost like a septic tank. That means it's full of E. coli. E. coli. So that's the name of the disease.
Okay.
So now we get into the next few graphs. This is done by one of my friends, a Japanese professor.
Yeah,
Dr. Fuji.
but
Now go back,
Professor, what I don't understand is why,
go back to the
Professor?
previous. Go back to the previous view graph.
Okay.
Okay, so in the middle, the small name, Dr. Tsuchi, and he is a professor at Kyoto University in Japan. So what he did, this research is beautiful, it's really beautiful. And it's an easy experiment, easy research, but I really envy him being so clever. So what he did is, he is doing the carbon implantation into the inner side of a silicon rubber, the first line. So the silicon rubber is a tiny tube, okay? So the tube is already showing about a few millimeter tube in the view graph here. The first is he cut the tube, then open the tube, therefore let the carbon ion bombarding the inner side of the tube. Then he sealed the tube together. Then what happened now is the tube become hydrophilic. That means the tube will love water inside. The water just flow inside easily, doesn't drop up. Then he put in two nerve. This is not nerve. This is really the spine of the rat. Okay, the spine of the rat are really inside this tube. Somehow they keep the mouse alive, and then trying to watch the spine grow together. Of course, they cut the spine. This is a very cruel experiment. They cut the spine and then separate the spine by a small distance, millimeter or so. And then inside this tube, trying to watch the spine grow together. Of course, the control experiment is without carbon implant. And then his sample is this silicon rubber also implanted with carbon. But he found out the growth rate, how fast the nerve grow together. The growth rate is improved by a factor of 4. That means they can grow together. So he worked together with the Kyoto hospital. So the intention is trying to see one day they can repair the human spinal cord. For example, during the traffic accident, quite frequently the victim broke his spine, and once the spine is broken, then the nerve is broken. Therefore, the locomotive on the lower part is missing, and therefore they have to be on a wheelchair. So suppose if one can really making the spine grow together, maybe the connection is restored. Therefore, the brain can still connect to the lower part of the body, and therefore one can free the patient from the wheelchair. Of course, they have to be rehabilitated and so on. So this is one part of research they were doing. Okay, let me keep going. Okay, I already mentioned this part. So this. Okay, I was skip many things. It's all related. So, but in this part, could you go to view graph 49?
49. Okay. 40.
The view graph 49 here shows very interesting pattern. So this pattern is really the cell. The pattern, first is they pattern the substrate. They pattern the substrate. And then by implantation. So this implantation is also carbon, because Dr. Tutsi is interested in carbon. He implant the carbon into the lines, those vertical lines, the carbon implant, the area. And then they try to grow the cell. The cell will prefer to the area where are hydrophilic. So that means the water will love to stick there. The water will be repelled by the other region. So the whatever water likes to be there, the cell will also like to be there. So a lot of cell nuclei around the implanted line. And so the line width is about 10 to 40 microns. So this is a non-nanometer type of research, but it's a large scale research and so on. So they show this kind of growth and so on. Next view graph. Okay. Next year, next one. T. Also, when they make the line width thinner, narrower, then there could be one or two nuclei, and they cannot squeeze too many of them so together. And they were thinking about this may have the neural network type of application. Next one. Next one. Yes, that's good. So you can also making square. Okay, you can making squares. You can making lines. You can making a tree shape. Therefore, the potential is there. So I have not followed them. So this was almost 10 or 12 years ago, what they were doing in the publication. Okay, so I think if I did not send this view graph making available, so I would like to ask T-Chain to really send this set to all the students. So therefore, my lecture is very, very hurry, and I also trying to just give you a feeling about what's going on and what's going on 10 years ago, and today it could be even more advanced and so on. Therefore, in a way, it's not just limited to the silicon, not just limited to the stainless steel or the alloys or metals and so on. And it could be done in polymer, it could be done in glasses, could be done in the biosystem and so on. First is open your mind. On the next session, I'm going to talk about it next week or some other time, is I'm going to go back to semiconductor and just emphasize on the doping, like junction formation, and some more detailed application, and also early day research and so on. So let me stop here.
Okay, thank you, Dr. Chu. And let me stop recording.